Hello, everyone. Thank you for joining us. This is our first YouTube live small cell smashers fireside chat. So if you will please hit subscribe and the alarm icon on your screens, that will turn on your alerts and then you'll be notified of all of our upcoming events. We are so excited today to speak to our own Dr. Misty Shields. All of you know who she is. She is the founder and inspiration behind all things Smashers. She is a translational medical oncologist at Indiana University, and that's why you see her in her football shirt today, as she is repping her team who are in the college national championship playoffs tonight. We will learn a little bit more about what translational oncology really means in just a moment. But Dr. Shields grew up in Texas. She holds a medical degree from the University of Texas Health in Dallas. She did all of her medical training both at University of Nashville, Vanderbilt University in Nashville, Tennessee, and then her fellowship in oncology at Moffitt Cancer Center in Tampa, Florida. She also holds a PhD from Texas Southwestern. And if all of this weren't enough, Dr. Shields is also a wife and mother, and today's her anniversary, and she has two beautiful children. So we are just so privileged to get to work with Dr. Shields every month and have her with us, as Dr. Roy said today, on the hot seat. I am also joined today, since Dr. Shields is our guest, my co-moderator today is going to be Dr. Upal Basu Roy, the Executive Director of all LUNGevity research. He oversees our scientific advisory board, our grant awards. He monitors and evaluates all research that LUNGevity is involved in. And he holds a PhD in molecular and cellular biology from the University of Arizona, as well as a master's in global health policy from NYU. He's also a dad to a beautiful son, and I can't say enough about what a great colleague that Upal is to work with. So let's jump right in. I know, Dr. Shields, that you have talked about this multiple times, but just for anyone who has not met you before and has not heard your story, what led you to become an oncologist, and especially to have a specific interest in small cell lung cancer?
Thank you, Ann, and thank you, Dr. Roy. And goodness, the honor is mine to work with you both, and the kind hearts and souls that you have towards all of our lovely smashers and patients and their loved ones is just you know, unprecedented. It's absolutely incredible, and I'm so grateful to work with you both and the LUNGevity team in this partnership. And so, my story for coming into oncology was, when I was a teenager. I was still obviously in Dallas, growing up. I'm the baby of five kiddos. And My dad, when I was in between the years of eighth grade and ninth grade in the summer, had a pneumonia that just wouldn't go away. And was ultimately diagnosed with a bronchoscopy with small cell lung cancer. And so, I was 13 at the time, and he was a veteran. He was at the VA in Dallas, and was diagnosed and quickly started on treatment. And as a family, we were a very close-knit family, and decided that we were going to keep this very close to our hearts and didn't tell anybody. And so went through his journey of almost a year and a half of fighting small cell, and he passed away in October of 2000, when I was 15. And so I knew that then, I would go to the VA with my dad and my mom to his appointments, and being there in the hospital and seeing the doctors and everything. I knew this was placed on my heart, that this is what I was meant to do. And so I'd known from that time. I didn't know how I was going to do it, but I knew that that was what I was meant to do, just a little over 25 years ago. And so fast forward, in the days of AOL, I, looked and found that one of the top lung cancer specialists had actually just moved to Dallas from the NCI, Dr. John Minna. And I reached out to him and said, "How do I become someone like you?" And he took a phone call from a teenager, and was kind enough. I guess I enticed his secretary enough to get him on the phone. And he said, go to college, get good grades, and come work in my lab sometime. And so I took him up on it and applied to graduate school only at Southwestern in Dallas and did my PhD with him in his lab. And Dr. Minna, if anyone doesn't know who he is, he's who saw all the patients at the naval branch of the NCI and all of the cell lines that we know for small cell, all several hundred cell lines were all his patients that he cared for, and established cell lines so that we could he discovered, P53 in small cell, RB in small cell, MYC in small cell, and did many of the pivotal early trials. And so his contribution to oncology is what he's the shoulders we all stand on. And so I worked with him and did my PhD with him, and in a collaboration way back in, late 2000s, early 2010s, we discovered NeuroD as the second subtype for small cell. But I really felt in my heart that I needed to see patients. And so applied to medical school, got in a little earlier than I thought, deferred a year, and then went on to do all my training, about 10 years more of training, and then became an oncologist in 2022. So that's my story.
It's incredible. It really is incredible.
And Dr. Shields, what a powerful way to honor your father's memory and legacy. Thank you for doing what you do. And you're also an incredible mentor to the next generation of scientists. You're already paying forward what you've heard from and learned from Dr. John Minna.
Yeah.
And Dr. Shields, I've sort of heard you introduce yourselves sometimes as a physician scientist.
Yeah.
What does that mean, being a physician scientist?
Yeah. So it means that you're kind of we wear both hats, so you wear this physician hat. And in my physician world I'm a medical oncologist, but you don't have to be a medical oncologist to be a physician scientist. You could be any oncologist, any physician specialty, a cardiologist, et cetera, a rheumatologist. But then on my other hat, I have a PhD and I have a research lab. And again, to be a physician scientist, you don't have to have a lab. You could be a collaborator. You can contribute to someone else's work. You could have clinical impact, or basic science impact, or somewhere in between. And so I really wear kind of two hats. And my I would say area of specialty is being a bridge between the clinical world where we see patients and the lab world where we study the science, being able to be in between, kind of that bridge or bench to bedside and back approach, being able to see patients with, and having the privilege to care for patients with small cell, but then going to the lab and studying small cell and being able to ask those questions that we see in patients, or the flip side, bringing our discoveries from the lab back to patients. So physician scientist really means that you're a doctor and you're a scientist, and that could really span many worlds. It could be epidemiology, it could be translational, it could be drug discovery. It could be many different things, but in my world, it would be all focused on small cell.
Dr. Shields, I think everyone on the Smashers, they know you as an oncologist and they don't hear a lot about your laboratory side of research.
Yeah.
So my next question is really sort of related to the research that you do in your laboratory and the research team that you lead. You talked about NeuroD1, and I think we've heard some discussions on the Smashers about the different subtypes of small cell lung cancer that's become quite mainstream now. And we're talking a lot about it, and NeuroD1 being one of those subtypes. How does your research relate to these subtypes of lung cancer?
Yeah, really, really great questions. So, the laboratory side, I started in August of 2023, about a year after I started on faculty. We're small but mighty. I have a graduate student, a postdoc, a resident physician who just got accepted to fellowship. I have a high school student who's phenomenal, and works on his afternoons after school here, and many summer students. And so we're small but mighty. So we have the typical lab space where you come into a laboratory. We have tissue cultures. We study all of these cell lines. We have about 50 of those small cell lines that Dr. Minna had established. And we do bench work, mouse work, and translational work, as well as, data interpretation, all of those things. In terms of the subtypes, I think that small cell is so unique in that we know that it's it is characterized but typically by two genes that are lost, and those two genes are very protective in normal cells. Those are p53 and RB, and those are kind of, if you think of them as the guardian of the cell, that are watching over the cell and making sure that no chaos, nothing that shouldn't occurs. And so it really is protective, or these tumor suppressors, they prevent cancer from forming. And in small cell, those are uniquely lost almost all of the time. p53 much more commonly lost, almost 100% of the time, and RB about 70 or so percent of the time. And so if you imagine, like, those are what I usually think of as if a policeman is watching a bank and he goes out to lunch, right? Someone can come in and have a robbery, right? Or something bad might happen because that policeman, that protective area is not there. And we can think about the subtypes as kind of those robbers, right? That they can do more harm because they're promoting something we don't want. And so if we think about those subtypes like NeuroD, ASCL1, PAL2F3, and a couple others that have been discovered, those are really these bad guys that can promote growth and ways to escape chemotherapy, ways to promote resistance. And so those are what we call transcription factors, or genes that turn on many other thousands of genes. And those are harder to control because it's a network, right? It's not just hitting one pathway. And so these subtypes are quite dynamic. They may change with therapy from a diagnosis to treatment, depending on what treatment. And we may hear things like plasticity, where something shifts from one area to the other. And so those subtypes, we don't know the right test for it. And I think that's what has been the hardest part from bringing it from the lab to the clinic is it a blood test? Is it a tissue test? Is it a something that's done at diagnosis or real time? Like, how often do we test for it? And I think those are the questions we just don't know yet the answers to. And so with the subtypes, there is a cooperative group that's looking at the SWOG is looking at this in the subtypes to see if we can target them potentially. And this is the first study to ever do it. And it's called PRISM, and it should be launching. Dr. Ann Chang is the principal investigator, and it's going to say, if we test the subtypes, can we stratify our first-line therapies based on those and help precisely target small cell lung cancer? So that should be launching. I think they're going to accrue maybe like 700 patients. It's a large study, a very big initiative to really answer this.
Dr. Shields, I believe that Dr. Chang was also a guest on one of your Smashers discussion, and your analogy of using the policeman for TP53 and RB, and then robbers for the transcription factors like NeuroD1, that's one of the best explanations I've heard about what's happening in small cell lung cancer. And sort of following up on this theme of subtypes, you got an ASCO Career Development Award last year, and that's focused on this protein called ISL1, which is a new protein which most of us have not heard a lot about. Can you tell us a little bit about what research you're doing on this protein and how that's going to impact clinical research?
Yeah, thank you for highlighting it. So this is a really cool story, and this is the power of one patient, right? So when I was a fellow, I had the honor of training at Moffitt Cancer Center in Tampa. And one of my favorite people in the whole world, one of my clinical mentors, Dr. Michael Shafiq. And I had the privilege of seeing patients in the clinic with him and learning his expertise and his mentorship as well for our research and clinical guidance, and helping me shape my clinical care I practice today. And with that, we had a lovely patient who was on a drug called lurbinectedin. And it is FDA approved, was FDA approved in 2020 for small cell. And now, as you've probably heard, in the fall, in December in October was FDA approved for maintenance for first line extensive stage. And so this drug is being widely used. It's a chemotherapy drug given every 3 weeks. What we knew about this drug is that most patients receive this therapy and maybe get 3 or 4 rounds, and scans might show things are growing. And we call that progression, meaning that cancer has progressed or there's a new spot, and it's not giving us the result we want, so we need to change therapies. Well, in this patient, he was on therapy for almost 2 years and had a complete response to therapy, had no evidence of disease with extensive stage, including had a history of a single brain met that was treated with radiation. And he's actually been off of therapy for almost 3 years with extensive stage, basically in complete remission. And so I wanted to know why. Why did this patient do so well? And so as part of that, I wrote up what we call an IRB, or a regulatory protocol, meaning it's a scientific protocol to say, this is the research I want to do. This is what I'm going to do with this information. This is how I'm going to safely protect the information. And this is what I'm going to learn from it and study. So we have to submit these types of protocols before we do a lot of research, anything handling patient specimens or anything involving patient care. Like getting into a patient's chart and looking at any variables or looking at their samples or studying them. So we have to have permission by our regulatory bodies to do that. And those are called IRB, or institutional review boards. And so I wrote a protocol and we said, let's look at some of the biomarkers that are being studied for lurbinectedin. And one of the most exciting ones was something called SLFN11, or SLFN11. And there was some data with SLFN11 and PARP inhibitors suggesting maybe those help patients improve response to PARP inhibitors. So we said, let's look at that and see. Well, unfortunately, that didn't pan out, but we had a plan B that we built into the protocol to say, let's search and find, if that doesn't work, what will? And so we took all of these patients. We had about 30 patients at Moffitt who had received lurbinectedin, and we found the patients who had available samples that weren't exhausted, that had different responses. They were very sensitive, they were very resistant, meaning their cancer grew very quickly, or they were on therapy for a long time, some of these patients for years. And we wanted to study what made them different. And so we took these samples and we said, let's do proteomics, where we're doing all this analysis of proteins that the cancer might be expressing. And the very top hit of all of that proteomics was this protein called ISL1, or islet-1. And so we said, well, what in the world is ISL1, right? We were just as profoundly shocked as you were as well, Dr. Roy. What in the world is this protein? And so when we looked and we started digging in the literature, it turns out that ISL1 is actually a transcription factor, just like the other subtypes. It's required for the pancreatic development, just like the other subtypes. And it turns out that it's expressed in small cell lung cancer.
Wow.
So we asked, perhaps maybe this is a subtype that's going to predict patients that respond to lurbinectedin.
It's like the fifth robber.
That's the fifth robber. And so we took it back to the lab. We confirmed that it was in those patient samples, right? You never want to just go blindly. So we did confirm that it was expressed by a different method. And then we took it back to the lab and we said, what is it doing? And so we characterized cell lines that had ISL1 and those that didn't. And those ones who did were very sensitive to lurbinectedin.
Mm-hmm.
Those that didn't were not. And when we looked at it, we could find what was it controlling, which are other genes called SOX5, 6-1, all these genes that are in the pathway that were also in our proteomics hits. And so when we take it away, the cells die. And when we look at the other subtypes, we don't see that it's switching to another subtype. And when we look at the actual ISL1 protein with lurbinectedin treatment, we see it go dramatically down, meaning that this is a biological target and it's important for lurbinectedin. And so with that, we want to know, what in the world is it doing in small cell? Why is it important for lurbinectedin? Our running hypothesis that we're working on now is to understand if ISL1, which binds as a transcription factor, is cooperating with this protein called SOX5, which the SOX family in this whole pathway bind the minor groove of the DNA and they bend the DNA open. The minor groove is one of the two parts of the DNA, the two helices, that it's the smaller groove that it bends open the DNA, which is exactly how lurbinectedin works. It works on the minor groove of the DNA. So I've hypothesized this protein is letting more drug get into these cells, and that's why those patients have more sensitive disease and have these success stories. So again, we are really still trying to understand this work. It has been accepted for publication just this last month, but we're still trying to understand what in the world does this mean? How common is it? Who has it? Does it matter with the beginning of treatment? Does it change over time? And is this something we can take to patients to say these are the right patients for lurbinectedin versus tarlatamab versus another trial with like an ADC? And so the idea is to bring this to patients, which is the foundation of our Conquer Cancer ASCO CDA award.
Wow.
Dr. Shields,
That
can I just
is
say how cool this is?
Yeah.
This is
exactly.
just amazing and a perfect example of sort of bench to the bedside and back to the
Yeah.
bench, that Anne was
Yep.
talking about earlier on, translational research. This is such a great example. And a fifth target for small cell. Wow, I
Yeah.
mean, this is amazing. Thank
Yeah,
you,
and
Dr. Shields, for
it
your
all started with
curiosity
just
and
one
perseverance.
one patient. So, one patient
Mhm.
matters. And for anyone who might be a researcher or an investigator, right? If you have something that's unique, those needles in a haystack, follow it, learn
Mhm.
from it, and then we can apply it. And so, this really started with one patient, and then hopefully will help impact many.
This is for our general community, this is probably sounds like very complicated science. Dr. Roy and I are science geeks, so we're completely fascinated by all of this. But when you really boil this down to our community, where do you believe this is leading us?
Yeah.
And how impactful do you expect this to be in the coming years? Because we've really had some exciting years here in small cell lung cancer that certainly in my professional lifetime is very new. So give our community a little sense about where you really think this is leading us and how exciting this could be.
Yeah. So this is such an important point. So, historically, small cell has always felt to be like a one size fits all, right?
Mhm.
Everybody gets carboplatin and etoposide, cisplatin and etoposide. And it was always felt to be this homogenous disease, and that couldn't be more wrong. It couldn't be more wrong. Small cell really is a unique um, type of cancer, and each one should be treated uniquely. And I think this helps bring that precision medicine or personalized medicine to small cell lung cancer, just like we see with patients who are on tarlatamab, right? The DLL3 bispecific T cell engager. It's such an amazing drug and can be a success story for some of our patients, but it's not for all of our patients. And those patients who don't receive benefit, wouldn't it be great to know who those patients are going to be before you ever get that therapy? And so that's where this role of biomarkers of having something that suggests with great confidence this is going to work or not work. And so that's the idea behind this work is, can we have a predictive biomarker to say you are going to respond with great confidence or you're not going to respond, and maybe you should try a different drug instead. And so, with small cell, we haven't had that yet. We didn't see with non-small cell, we saw PD-L1, we see EGFR, we see KRAS, we see ALK, ROS1, NRG. I could go on, MET, etc. And these therapies really revolve around having a biomarker or a gene that is broken, altered, driving this cancer that we can target with a targeted therapy or immunotherapy. But in small cell, we haven't had that just yet. Even, with immunotherapy, we don't see that PD-L1 or that score matters in small cell. And with tarlatamab, we don't see that DLL3 matters. So this is a disease that needs biomarkers, and I think we are going to get there. And my hope is that this research represents one of those potential victories, to say this is the biomarker that if you have this is the right person that should go on lurbinectedin, versus if you don't, maybe you should consider something different, because every decision we make for patients has impact, right? It's time, it's money, it's time toxicity, it's financial toxicity, it's side effects, it's, sitting in infusion and getting scans, and every decision we make has a ripple effect. And so we really want to be mindful that we make the right decision with as much evidence as we can to predict that, and give great confidence, as much as we can with scientific evidence. And so this is my idea, is that we can find more of these biomarkers to help patients make those decisions.
Yeah. Yeah. It's amazing how far we have come, and how exciting this truly is for a community that, as you say, has really only had one set of treatments for more than 30 years. There were two drugs, and after that it was sort of a toss up as to what might work and what probably wouldn't. So this research is just so fascinating. And what's amazing to me is how you balance this. Like, what is a typical day in the hospital or your office at your lab? What does that look like? Because how do you balance this research with actually seeing your patients? And I know from working with you, and I know from hearing from your patients, that you are definitely one of those who sits down and spends time with your patients and talks with them and makes sure that all of their needs are met. So how do you balance this?
I think this is a team effort, right? I can't do any of the research I do alone. I have a fantastic team in the lab, as well as my translational team, who works with, interacting with patients, and patient-facing care, or helping to, you know, document or collect all this data as well. So this is really a team effort. So I can't take any, any and all of the credit, here. I have a lovely team. But I think, my biggest, one of my biggest philosophies is be wherever you are, right? And I think so many times wherever we are is distracted, you know, um, and, being present, being wherever you are, if you're in the clinic, being there with your patients, you know, being focused on them and not a computer, right? Being focused, in your lab, being there present in the lab and focused on research, and so many of the things, all the noise that surrounds us, in this new day and age, I think can be quite distracting. And so really being present and giving, our focus to whatever it is in front of us, I think is helpful for me to balance. And again, I can't do any of this without my amazing teams, especially my clinical team, my research nurses, my clinical nurses, coordinators, infusion, scheduling, um, and all of our staff, that helps to make things run on a daily basis. And I think that it really does. This is an army of people. I just get to help be a part of and lead.
I think that's a really important point. And we've talked about this, and in our other fireside chats, and we've really sort of dissected what a multidisciplinary team looks like.
Mm.
But I do think it's a really important point that you make, Dr. Shields, that it really is a team effort. It's not a singular person. It's not, and it goes back to that word translational that we were talking about, and what that really means about how you are present in the lab and take what you're learning in the lab and then bridge that over or translate that to your patients. It's that's a really important point.
I think so. And there's, all the things that we do are so designed for distraction. I think that, it's like every ad, every like a little pop-up and notification and alerts. And, I think it's, pushing back, hacking back on all those noises, really being present with your patients, with your loved ones, with your teammates, your colleagues, being there, and being present. I think it has helped me to balance.
And that's advice that we can all take to our respective lives, Dr. Shields,
Yes.
personally and professionally, just being present. Um,
Yeah.
I wanted to sort of pick up on Ann's point about translational research, sort of, bridging both sides. And I know that, we've not talked about this particular piece a lot today, but again, just sort of reminding us ourselves that there is also a subset of patients who've had non-small cell lung cancer, and once they get treated with a tyrosine kinase inhibitor, that cancer switches and becomes small cell. And I do know that that's also one of your research interests, and you do have, I think, a clinical trial, I think, in the works in that space, sort of really to serve that group of patients who go from non-small cell to small cell. Can you tell us a little bit about this new trial that you are conceptualizing?
So, thank you for highlighting that. It is what's called an IIT, which is an investigator-initiated trial. So many of the trials that we see come from different avenues. They may be investigating a drug, or a compound or an agent, et cetera, or an intervention. And that might stem from the pharmaceutical company, that might stem from an investigator or a cooperative group or other, agencies like regulatory, like the NIH or the VA. And so this was a study that, because I saw, there was an area of great need, and these patients they're always lovely, and they are quite isolated, and I felt like needed um, needed some guidance, and needed a study that focused on them, needed something to guide their treatment, because there really wasn't, because it's such a rare population. As mentioned, it's patients who have non-small cell with EGFR. Sometimes it can be ALK or other oncogenic drivers, but then because of how effective the therapy is, the way that the non-small cell escapes is it changes to small cell. And so those are our patients who may have transformed small cell, or, uh, we call it TSCLC, or transformed small cell. And so this study is looking at work that's built upon, one of our amazing speakers that we had, Dr. Chaparna Sen, who I know, I think you helped me co-moderate that one, Dr. Roy, who is a phenomenal um, basic and translational researcher in small cell. And this is based on her data that she generated in the lab and on patient specimens, that potentially lurbinectedin and osimertinib, the EGFR therapy, work well together to really target this transformed small cell. And so the idea is to study this in a trial setting and see if it really does work. There are, thankfully, unfortunately, some other studies that are being used in transformed small cell, like tarlatamab is being investigated, finally, for patients with transformed small cell. And through other investigator-initiated or IIT studies. I think Dr. Alyssa Cooper has one at Dana-Farber that she's launching as well. And so this is a lovely patient population that has been excluded from so many trials, and it's time for us to study these patients and help get them therapies that are right for them.
Thank you, Dr. Shields. And I promise I have just one more question because Anne knows I can
Yeah.
do this for a long time. My last question is really about something we've not really talked about, but I do know is also another big research interest of yours, which is using non-invasive tools to monitor people living with small cell lung cancer, for example, blood biopsies. Can you tell us a little bit about what
Yeah.
research is happening in that area? And I know you're really passionate about this. I'd love to hear a little bit more about what you're doing here.
Yeah. Thank you for highlighting it. So this is what you're highlighting is really this liquid biopsies. Our patients may hear this. Liquid biopsies can be for kind of commercial use or research use. And so they may hear commercial use of like different companies like Foundation One or Caris or Guardant, Tempest, these different companies that have commercially available, regulatory approved tests that can be done to look for certain genetic mutations, how much cancer might be in the DNA. And those are really on a set panel of a certain number of genes that might be cancer causing or related to cancer. This is a program I started in, goodness, April of 2023. So we're in our third year now, and it's a program called MACROS, which is our liquid biopsy program. It's an acronym for basically looking at mechanisms of acquired resistance for patients who have small cell lung cancer. And so what it looks like is that patients are approached by my team here at Indiana University, and they're consented and collected at baseline, ideally before they ever start treatment or they start a next line treatment. And then while they're on scans longitudinally, so over time. And so every time they get a scan, they get 3 tubes of blood, and then that gets biobanked for us to study. And so we've had almost 100 patients who have graciously donated and participated in this program, which means over 1,000 tubes of blood. And this is a phenomenal altruistic donation of these patients to help us to understand small cell. And the reason is that small cell is very rarely rebiopsied. And the biopsies we get may not have a whole lot of tissue or information. And so this helps us to study it over time. It's minimally invasive. It's tubes of blood that we're getting, so many tubes of blood anyway when we're at clinics. And then we can study them and look at DNA and RNA and protein and subtypes. And we can look at by stage or any toxicities. Let's say they have cytokine release syndrome or ICANS, or they have a perineoplastic syndrome like Lambert-Eaton or SIADH, or they have different treatments like tarlatamab, lurbinectedin, chemoimmunotherapy, immunotherapy and lurbi, immunotherapy alone. And we can study patients by what drugs, where they are in their treatment, and what their response is on scans. And so this is a huge team I have of my regulatory staff, my research staff, but then I have a phenomenal team of our lung cancer database. That's about 10 different, very dedicated medical students, residents, fellows, and faculty that really are looking at this. And as well as my research lab, my postdoc leading this effort on our side. And then collaboration with a phenomenal researcher, Dr. Max Steen, who is a liquid biopsy giant, really guru, to study these and say, the patients who responded well versus the patients who didn't respond well, what makes those patients unique? Or the patients who are going to progress, why? What is it that we need to target to make them not progress? And so our current focus right now is on tarlatamab. To find a biomarker. We have about 45 patients who generously participate in this program and are on macros, so we can follow them. And not all patients, as we know, not all patients respond to tarlatamab. And so unfortunately, many of our patients have progression, and we need to figure out why and learn from them and help the next generation of patients. And so this is, my patients are so kind to donate, and they know that it may not help them, but it might help the next generation or the next patient that comes in. And so these discoveries really impact the greater good for patients with small cell. And so we have our initial kind of analysis on patients with tarlatamab, some really exciting things I can't talk about just yet. Maybe I'll be back at another Smashers Live. I don't know. I'll have to talk to the boss, I guess. And we could talk about those at a later time. But I think very exciting, and biomarkers, again, help guide the therapies to the right patients and avoid the patients who may not benefit and lose time and energy and investment to a different drug that would work for them. So we just want to help patients be as efficient and invest in their time and their efforts and as much as possible. And I think science can help us get there.
This is just brings to mind something that I don't think that we talk about very often. And we do a lot at LUNGevity in promoting clinical trials and having the patient voice in clinical trials. But I think one of the things that we probably don't talk about when it comes to clinical research very often is that not all clinical research is directed around a particular therapy. And what you've
That's a very good
highlighted
point.
in this liquid biopsy and just, everything you've talked about today in terms of these subtypes and looking for biomarkers, how important it is that for our patients out there, that if your provider approaches you and asks you for extra tubes of blood and those kind of things that could seem kind of annoying and like, what are you doing with this and how is this going to help me? I mean, you have really highlighted today how important that aspect of clinical research is and how important it is that we learn from individual patients. So I really thank you for highlighting that and helping our community to understand that not every protocol, not every research project that you might be approached for actually has anything to do with your therapy. Maybe just trying to learn more about your disease so we can move treatments forward.
Yeah, this is such a critical point because not all studies, like you said, are interventional. They don't have to have a drug. If you even look on clinicaltrials.gov, there's an option to say non-interventional versus interventional for these resources as well. And to see if there's something that's open or available because it is so helpful. I know back to the PRISM study, with Dr. Ann Chang and the subtypes, they're as part of that study going to do probably the largest comprehensive analysis for genomic testing in that study. So it's going to be a massive resource for investigators for future translation because they'll study all of these patient samples comprehensively as well. So it may be even rolled into the studies that you're on and you just, maybe on the next page or a couple pages down on the consent form where you see that they talk about genetic testing or tumor testing or different correlative analyses. And so it is so helpful for us to understand because for so long we didn't know and we only knew what was at the beginning of small cell at diagnosis and not how the disease might change over time. So this really helps provide insight.
Dr. Shields, I do believe there is a question from our chat for you about Lurby, and so maybe you want to take that question?
Sure. It looks like, let me see here. The question is about, is it only in line one for lurbinectedin? So, how the original basket trial that led to the accelerated approval of Lurby was designed was that it was for at least relapse small cells, so second line and beyond. And that showed that it worked in about 35% of patients if patients had what we call platinum sensitive disease, meaning they had been off of platinum-based therapy and on immunotherapy only for really more than 6 months. Those patients might do even better on lurbinectedin, meaning almost half of patients respond. So if it was initially sensitive to one type of therapy, it might be even more sensitive to Lurby than kind of the general population. And so that led to the FDA approval of it being second line and beyond. And then we saw the IMforte data at ASCO this past year, our big oncology conference in June of 2025, that showed that Lurby in the maintenance, so with immunotherapy, improved survival and to help the disease from progressing or growing. And so then it was uptaken as a first line maintenance. And so typically if you have it in one area, we don't want you to have it in a second area. Like if you have it in maintenance and it stopped working, we don't want you to have it in the second line or third line because you don't want to keep doing more of the same if it stopped working. But typically it would be either in the maintenance for many patients who might be now pursuing first line therapy, or in the second line beyond, second or third line, et cetera. But again, we don't usually re-challenge with it if it ever stopped working.
And there's a second part to that question about ADCs, and the question was asked, what is an ADC and when are they used?
Very good question. So ADC is an antibody drug conjugate. So I always think of this as like a biological missile, or a Trojan horse that has an antibody and a chemotherapy that's attached to each other. And those antibodies go recognize something on the surface. It's like, I've heard the analogies of saying it's having a addressed envelope, like a letter, and then that is the address that antibody is going to recognize the address that's on that letter and be sent to that mailbox. And inside that mail is going to be, this chemotherapy payload. And nobody would want to get that, open that mail, I guess, in our scenario. But it would be allowing this chemotherapy in the small cell to come in. And so it is like a Trojan horse being welcomed in by the small cell because it's attached to an antibody on the surface of the small cell. And so those antibody drug conjugates, these kind of chemotherapy warheads, can be, they were designed against multiple targets. One is something called SEZ6, another drug called IDXD or B7H3. Another is TROP2. Actually, there's one against DLL3, the same target of tarlatamab. And there's many others that are in the works and being designed. And so the idea is you can recognize something on the surface, target it, and then deliver a high dose of chemotherapy directly to that cell, because it's ushering in the chemotherapy, unassumingly, because it's bringing in this antibody, and lo and behold, attached to it is chemotherapy. And so many of these drugs have been studied in small cell. I would say IDXD and the TROP2 antibody drug conjugate sacituzumab govitecan, probably the most well studied so far, farthest ahead in development. They all have, those two have breakthrough designations by the FDA, but not full approvals yet. We actually heard at the end of December, kind of flew under the radar, that IDXD is on a partial hold by the FDA because of interstitial lung disease, which is pneumonitis. So they're investigating. They had a few patients that had a toxicity that could be potentially life-threatening. And so they are studying that to make sure they understand that risk, before it would be potentially approved by the FDA. And so these are really exciting drugs. They have high response rates, meaning that a large number of patients who receive this drug are going to have benefit, and that it works very quickly. So usually the first scan or 6 weeks in, we'll see shrinkage in multiple areas, including anywhere in the brain.
This is another question that says, does doing the genetic testing limit treatment in some cases?
Let's see.
Small cell at the moment, it helps to have many options if you have an oncologist willing to think outside the box.
Yeah, so I think Ann and Dr. Roy, you would agree. I think the thoracic oncology world is a lovely community, right? I think that the oncologists who care for patients who are impacted by lung cancer, and particularly in small cell, are just so kind and just phenomenal. I have phenomenal collaborators and colleagues all over the world. I think you guys would agree, right?
Absolutely.
Yeah. And so I do think that many of the oncologists try to advocate as much as possible and think outside the box. And, I do think this is where power and second opinions and third opinions and getting other doctors' insights, if you feel like you're not being supported, definitely doing so. But in terms of genetic testing limiting options, I can't think of any particular examples. I don't know if you all have, but I think with With small cell, in 2022, we wrote it into the guidelines for our national guidelines with the NCCN, or the National Comprehensive Cancer Network, that patients for therapeutic or diagnostic dilemmas should get and be considered for genetic testing, like a liquid biopsy or tissue biopsy, for understanding the genes for small cell lung cancer, because that might open the door to a trial that we didn't think of before. And every so often, we'll see different genetic alterations that we might already have a pill therapy or a targeted therapy, or it might open opportunity for an investigational agent like a clinical trial, or just help us understand why the disease is acting in a certain way. Like, let's say we don't see an RB mutation. That might help us to understand if the cancer is not responding to therapy, or if, let's say, immunotherapy didn't work well. So I think it is helpful to understand the biology. It shouldn't limit options, but hopefully expand them.
Well, we are coming close to the end of our time. This has been absolutely fascinating. I really just want to ask you one more question, Dr. Shields, and that's, we all talk about New Year's resolutions and goals, and I know that our Smashers community is just very near and dear to your heart. And are there any things that you have aspirations for or goals that you would like to see us all find this year in our Smashers community?
Yeah, we are so blessed to have our Smashers, all of our patients and their loved ones, their care partners, who are so dedicated to supporting and encouraging and uplifting each other. And I'm incredibly grateful. And I think it's just such a kind and supportive community. One thing I think would be great to get us all together, right? To get us in the same room. And I think, opportunities like the Hope Summit are the way to see each other, to give each other hugs, to see each other face to face. Zoom is great, but the human element is there with encouragement and laughs and hugs and seeing each other in person. So I think getting each other together would be great. I think that, I think there's more advances for our patients to come. I'd love to make sure that all of our patients have equal access to therapies. I know so many of our Smashers are overseas, Australia, the EU, and other continents, and that may not have fair access to some of the drugs because they haven't been approved yet. And so I'm hopeful that some of these drugs like tarlatamab will get approved and be able to be accessible for all of our Smashers, because I know that's something that really hits home for me, to see some of our Smashers not be able to access that's really tough and just not fair. And I think, um, but making sure that we advocate for ourselves, our loved ones. I know that so many of our Smashers are doing that, and I encourage you, keep going. You got this. Keep smashing. If you feel like your voice isn't heard, go find another person to talk to. And really, if you're ever stuck, you ever need help, you never ever need an ear, we're here to listen in and support you.
Absolutely. Absolutely. And just to follow up on that, we are planning a Smashers live event, which will happen on the Friday of our Hope Summit weekend, the first weekend in May. It will be fully dedicated to our Smashers that day. It's brand new. We have not done this at LUNGevity before, and we are super excited. And our registration will be opening very soon. So you can check the LUNGevity website. We hope that by the 1st of February that all of our registration is open and you can register there. Dr. Shields, as always, this has been a wonderful, wonderful conversation. Just so much exciting research going on, and so many new opportunities coming for our Smashers community. This is just amazing. And Upal, my friend, love co-moderating anytime with you. You add such a brilliant scientific voice to all of our conversations. So I appreciate so much you putting this on your schedule today and joining us. Again, we are switching over to a new format of YouTube Live. So please do hit those icons and subscribe on your screen today so that you will get notifications. As Dr. Shields mentioned, we are always here for you. And if you have not joined our Smashers community online on the Small Cell Smashers Facebook page, please do so, because Dr. Shields does monitor that and moderate that herself. And often, if you are looking for a second opinion or a clinical trial, she can answer those questions for you. Please join us again in February. On February the 6th, we will be joined by another one of our dear friends and medical oncologist in the thoracic world, Dr. Eric Seeney from MD Anderson Cancer Center. And until then, Happy New Year, and thank you all